Alright, last section in chapter 10 is going to be on regulating the cell cycle. Here's a list of objectives. Remember, read over them. Make sure that you can answer these questions when you get to the end of the lecture to assure that you have full understanding and comprehension. Okay, so there are genes that are in charge of cell division. Cells are not constantly dividing. What would cause them to divide? Well, in case of an injury, for example, if there is a cut in your skin, you have damaged or missing cells now. And so it's going to stimulate cell division from nearby cells to allow that cut to be healed and to replace those cells. Okay? Once that repair has been made, then the division is going to turn off. So that's one example of when we can use or when we turn on cell division. Okay? Depending on the type of cell and its need, it's going to have different rates of division, meaning how often it divides. For example, skin cells, they divide once an hour, but think about it, you lose a lot of skin cells throughout the day. Then there are other cells like your liver cells. While we know your liver is regenerative, it takes a long time for it to regenerate part of itself. They divide about once a year. And then there are some cells like in your nerves and in your muscles that do not divide ever again once they form. And so if you remember from the cell cycle, then that would mean that they are constantly in G sub 0, where they have no intention of ever entering division again. So therefore, think about the effects of an injury to nerves versus an injury to your skin. Much more damaging and long-lasting to affect your nerve cells. So we first discovered that there was some sort of control over the cell cycle not that long ago, actually in the 1980s. And we did an experiment, and this is how the experiment goes. Okay? Here we have a cell that's in the middle of mitosis. And what they did is they stuck a pipette in here and they sucked up some of the fluid. Then they took that fluid and injected it into a cell that was not dividing. Okay? Once that cell was injected with the cytoplasmic fluid, they found that the cell then immediately started division. So the conclusion here was that there is some protein that is in the cytoplasm that tells a cell, turn on cell division, it's time to divide. Okay? That protein is called a cyclin because throughout the cell cycle process, the amount of cyclins that are found in the cytoplasm are going to increase or decrease. Okay? The closer we get to division, we're going to see a huge increase in the amount of cyclins that are found in the cytoplasm, and then it will decrease when the cell is not interested in dividing. So the job or the role of cyclins is to regulate or control the timing of your cell cycle. So they are proteins in the cytoplasm that control the timing of the cell cycle. So let's take a look at some proteins or some regulators that are found inside a cell that are going to say division is okay. As you go through cell division, there are lots of little steps, and so the cell wants to make sure that it is able to progress from one step to the next, having accomplished everything it's supposed to do. So it's set up these little protein checkpoints. Okay, for example, here's one. Remember G sub 1, the G1 phase in interphase. There's going to be a little checkpoint here. So it's sort of like a protein is standing there and he's holding a clipboard, and what he is looking for is he's checking to see if the cell has increased in size enough, if we have enough size to the cell. Okay? If it has, then he says, okay, continue. So we move through G1, going through S, we know that's when our chromosomes are copied, and we get down into G2, the organelles. We're just about ready to divide, and then boom, we hit another checkpoint. And this protein is there to assure, have all of the chromosomes been copied properly? If not, the cell does not move forward. If yes, okay, continue on. So then here we are again. We're going to go into mitosis. We're going through prophase, metaphase, and stop. And there's another checkpoint here. And here this protein is checking to make sure that all of the sister chromatids are properly attached to the spindle fibers before anaphase would pull them apart. Okay, so they are protein regulators that are responsible for controlling these parts of the cell cycle. There are some external regulators as well, and so what's going to happen is a lot of times these external regulators come from outside of the cell. So the internal regulators were found within the cell that was dividing. The external regulators are receiving signals from outside of the cell. The first example would be growth factors. Okay, so these are another form of a protein that are released from neighboring cells, and they act as signals. And it's very important during wound healing or embryo development. The cells around you release signals in the form of molecules to say, hey, we need to speed up cell division, or hey, we need to slow down cell division. Okay. Another example is density-dependent inhibition, okay, or what we call contact inhibition. I would write that on your notes if I were you. Contact inhibition. So these signals, they're going to prevent any sort of excessive growth and keep tissues from disrupting one another. Okay, let me see what's on the next page here, if this is the picture I want. Yes, okay. All right, so looking at this petri dish here, you'll see that we have sort of 3 separate isolated groups of cells, and they are actively dividing. Okay, let's see what happens here. We go to the next page. Okay, as we go to the next picture of the petri dish, we see that the cells have divided in such a way that they grow until they come in contact with each other. So here, the signal they receive is the physical contact with another cell. It's sort of like, you know, you're free to stretch out all you want until you feel walls around you or you feel other people around you, and then you can't stretch any further than that. So they grow until they maintain contact with another cell. So that's how we get just a plate full of cells, and they're not, you know, spilling over the sides or anything like that. So then the scientists said, okay, well, are they going to regulate themselves again? If we remove the cells in the middle, will they start growing again? Yes, they will. Okay, and so these are newly dividing cells. The cells back here, all right, these guys, they do not need to grow anymore. So their cell division genes have been turned off because they are still completely surrounded in contact with other cells. But these guys in the middle, there's space to grow. And so because of that, they will turn on those cell division genes. How long will that last? Until they then also maintain contact on all sides with other cells, and then they will be shut off. So the important thing that this slide is telling you is that is the contact surrounding other cells that is going to help control cell division. So this is a form of an external regulator. Okay. Unfortunately, some cells do not respond to all of these signals or controls that we've been talking about. And when they lose that control, then that is when cancerous cells can develop. And here are examples of cells. Okay, let me just show you with the pointer here. All right, these two are cancer cells on either side. These guys, these are white blood cells trying to attack them. Okay, but these are cancerous cells that have been developed from what are called CHO cells, or Chinese hamster ovary cells. It's just a type of cell that is used a lot in research because it's very easy to work with. But these cells are now considered cancerous. So cancer is a disease of the cell cycle, no longer able to control their cell division genes. So there's been a mutation in those genes. Okay? First place that we see this, when we have cells touching each other under normal situations, that would cause them to signal to stop all cell division. But here, when they come in contact with other cells, they don't stop, they will just keep growing. And so that is what ultimately makes a tumor. And you'll see we have this uncontrollably growing mass of cells here, and they're all just piling up on top of each other. It's important to note each one of these cells is abnormal in that it contains the mutation for those improperly formed cell division genes. So they are not doing their job anymore. Okay, so let's talk a little bit about tumors. Honestly, if I had my way, we would spend weeks talking about cancer. I think it's a very important topic. In addition to that, I think it's a great way to teach about cells and about genetics and about the environment and all these other topics. But alas, your Keystone exam does not put that much emphasis on it. So I'm just going to give you a little bit of extra information here and then we can move on. So we know a tumor is a mass of uncontrollably dividing cells. But the question is, when does that form the disease cancer? Okay? First of all, you can have benign tumors, and benign means not cancerous or not having a disease of cancer. The reason we call it benign is because this group of cells is relatively small, does not spread to other body parts, and it usually can be easily removed through surgery. So for example, the most common tumor that you guys probably interact with is if you have some sort of a mole or a beauty mark of some sort that you've had removed. Okay, it probably is benign right now, but it could turn cancerous for any reason. So that's why they have them removed a lot. But, generally, that does not mean you have cancer. You just have a mass of uncontrollably dividing cells there, and they have, formed a mass. And so the coloration comes from their intense density. They're all piled on top of each other. Where things get dangerous and where we start talking about cancer is when that tumor becomes malignant. Okay, this is considered a cancerous growth because of metastasis. Okay, big word here. This was in your objectives, talking about metastasis. Metastasis is literally when cancerous cells spread from one body part to another. Okay, think about it. What would be the easiest way for those cells to move throughout your body? Exactly, the super liquid highway you have running to every cell in your body, aka your bloodstream. So on the next page I have a diagram here. Okay, in this diagram we're showing up here we have a small mass. Technically it would be benign, okay, but all it takes is for that one little cell, or two cells or whatever, to leak into the bloodstream. Okay, so now here's this cancerous cell, and it is moving through the bloodstream. And let's say it travels to another organ. Let's say it goes, from the breast tissue down to the liver. Okay, when it was just in the breast tissue, it would be considered a benign tumor that was, could be removed through surgery. But now this little guy has gotten into the bloodstream and he's moved down to the liver. Well, as soon as he plants himself in the liver, he's going to start dividing as well. And so now we have cancer in two areas here. It's not just in the breast tissue anymore. It has now invaded another organ. So this would be considered metastasis, and therefore the tumor is malignant because it's spread through the body. And this is the beginning stages of the disease of cancer. Okay, another picture here just to show you the difference of malignant versus benign tumors. Okay, you can see in this picture, very small tumor, just sort of on the surface. Okay, now we're starting to get bigger and bigger. Okay, we're invading neighboring tissues, and this is an important concept here. Malignant tumors are generally not able to be removed by surgery because they have wrapped themselves around blood vessels. And so in order to cut the tumor out, you'd be cutting into your blood vessel supply, and that risks death from bleeding out. And so we would call these tumors inoperable. Majority of the time when you develop tumors in your brain tissue or deep within other organs, such as like your bladder or your ovaries, they wrap themselves in these blood vessels and they are therefore inoperable. So what causes cancer? Okay, because I'm sure you've heard all sorts of things in the media. Bottom line is it's something that has happened either that you inherited something or you were exposed to something in the environment and it caused a mutation. Okay, big importance here on the word mutation. Sometimes it can be spontaneous, and we're going to talk more about changes in the DNA in the coming chapters, but the idea that, oops, a mistake was made when we were copying the DNA during S phase, and, it just happened to affect a cell cycle gene that was, can no longer regulate. Sometimes it's a mutation that's caused by the environment. For example, carcinogens, mean cancer-causing agents. We know radiation, tobacco. There's actually a virus. It's called HPV, human papilloma virus. Its infection in your body causes the abnormal growth of polyps on the colon, and polyps are sort of like, I don't know if you've ever seen like skin tags, like there's sort of like a little growth on the side of, usually you see them on necks or like, under your arm or something. It's just a little bunching up of the skin that sort of sticks out, except that's now on the inside of you, on your colon, caused by a virus. That abnormal growth can lead to cancer, okay? Sometimes there is a gene error that you inherited from your parents, okay? But very important here, this statement, okay? Nice star around it here. All cancers are genetic, but not all cancers are hereditary. What does that mean? Well, basically, genetic just means affecting your genes. Okay, so every one of these things, whether it's radiation, whether it's coming from the environment because you're a smoker, all of those affect the genes and mutate your genes. But it does not necessarily mean you inherited them from your family. Okay, so that's very important to be able to distinguish. You do not inherit cancer. You can inherit some mutated genes that could lead to cancer, but most often you're going to get them from your environment. Okay, this is just a listing of different types of cancers. Usually I spend more time on this. We do a big project and things like that, but I want to keep moving because it's not considered that important. Who knows, maybe someday I'll write a special course on the study of cancer biology, and then if you're interested, we can really delve into it. But these are the names of all the different cancers and what parts of the body, what types of cells they can be affected by. Okay. Here's a list of all the different types of genes that are associated with cancer, and note that they all play some sort of role in regulating the cell cycle. Okay, whether they're a checkpoint gene, whether they repair DNA, whether they they signal to the cell when it's time to divide or not. Cellular senescence genes, that's meaning deciding when the cell should be going into apoptosis or not, if it's diseased. All of these things, again, big take-home message, cancer is related to the genes that affect the regulation of the cell cycle. Okay. Just looking at two genes, we have oncogenes and we have tumor suppressor genes. Oncogenes are like the yay, cells grow. Okay, their normal job is to stimulate cell growth. A tumor suppressor gene, his normal job is to stop cell growth. So under normal conditions, you have oncogenes and tumor suppressor genes being turned on and off in response to each other, so that, we could stimulate a little bit of growth, and then when we're growing too much, we turn them off and turn the tumor suppressor genes on. Okay. The problem is, if there's a mutation in an oncogene, then it's like saying, hey, let's grow and grow and grow and grow and grow, meaning divide over and over and over again. And that's going to lead to that accelerated rate of cell division. So it would be sort of like tying your foot to the gas pedal of a car and then tying the gas pedal to a really heavy rock. It's like, woo, the gas stays on, and it's just go, 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 divide, divide, divide. Or you can mutate the tumor suppressor gene, okay? So when a cell receives a signal to say, we need to slow cell division, it says, no, I'm not going to slow cell division down. So that would be like trying to apply the brake in a car, but having the brakes broken. So both of those are mutations that result in accelerated cell division. Okay. Cancer is a very complex disease, and a lot of people are personally affected by it. The statistic is that by 2014, it could be even up to 1 in 4 people will develop cancer in their lifetime. We are certainly very close to that at this point. There are many different causes for it, from the environment, things like that. It's a step-by-step process. You have to accumulate all of these mutations together. But one thing they have in common is they've lost the ability to control their cell division rate. That's very important. Your book specifically talks about a gene that has been sort of labeled as a cancer hotspot. It's called P53. Just stands P for protein, and its job is to repair damaged DNA. All right, so let's take a look a little more closely at this. So on the left side, normal P53. I sort of think of it like a policeman, okay? So P53 is called to a DNA damage site. Let's say radiation changed your DNA and it's been damaged. The P53 gene binds to that site, so it's sort of like the policeman arrives at the scene of the crime and says, everything stop, okay? So it stops cell development, no more division, and it either allows the cell to repair itself, or if the repair cannot be made, it tells the cell to program itself for death, or apoptosis. Now, if that P53 gene is mutated, here's what happens in response. Okay, we have the same damage somewhere else in the DNA, okay, but this time when the policeman gets to the scene of the crime, he can't do anything because he physically can't bind to the site of the damage. And going back to chapter 2, it's a protein, so what happens that makes a protein defective? The change in shape has occurred, so it cannot physically bind to the site, which means it's not going to stop the cell from developing. So what happens is the cell is going to continue growing and dividing in an uncontrolled manner. Now it is passing on even more damaged DNA as it divides through mitosis, and what we get is the development of many cancerous cells. This is just a diagram to sort of reinforce that. If you want, you can pause here and see if you can walk yourself through it. Look at what happens when P53 is able to function under normal situation or when it's mutated. Okay. Basic treatments right now, since we can't do anything to prevent cancer, chemotherapy, radiation, and surgery are the treatments. In chemotherapy, we use a liquid drugs, or we use pills. It doesn't matter either way. They're drugs that target cells in the body that are dividing, okay? So that's going to target your cancer cells, obviously, because they're dividing uncontrollably, but there are also other healthy cells in the body that are dividing. For example, your hair is constantly dividing, and so that's why when people have chemotherapy or radiation, their hair falls out because in addition to killing the cancer cells that are dividing, it kills the good cells in your body that are dividing. Also, a lot of people get sick during chemotherapy and radiation treatment, and that's because the lining of your digestive system is constantly dividing, and so those cells are being killed, and that that can cause a lot of the nausea side effects. Surgery does not technically have any side effects, and that is just a way that we can remove isolated tumors. Again, very important that those tumors would still be considered benign, or at least in a very small, confined stage. Okay. And the last thing here just to go over is looking at risk factors. Okay. Age is a huge risk factor, and we're going to be doing an activity on this. But age, as we increase in age, it increases our chance of developing cancer. And it's not because, old people suck or anything like that. It's just a person who is 5 has had, how many chances for their cells to divide. Whereas a person who is 50 has had so many more chances for their cells to divide. So it's a strict rule of probability. Just the fact that there are more abilities or more chances for division by the time you're 50, that increases the chance that one of those divisions went haywire and a mutation has occurred. They found that physical activity is strongly linked to it. Okay, so maintaining an active lifestyle, not accumulating too much fat. Exposure to radiation, for example, fake bakers, gotta be really careful. Also, just if you're going to play outside in the sun a lot, radiation is definitely one of those factors that can cause cancer. Alcohol and tobacco, because of the negative effects they have on your immune system, and also the fact that they are technically poisons to the body, they can cause mutations. Some of these other chemicals, benzenes, so this is the reason why you are not supposed to freeze or heat plastic water bottles at all, because the plastic has a chemical in it that could cause cancer. And we talked about the virus here, HPV. Bacteria causes hepatitis B and hepatitis C. Both of these infections can cause an issue with the DNA, and depending on if there's a mutation in a cell cycle gene, it could develop to cancer. There are some cancers that have stronger family history ties than others. For example, breast cancer is like 85% based on your family inheritance, and I believe prostate cancer is the same way as well. So that was just sort of a quick snapshot on cancer. We're going to spend a lot more time on it talking in class. I will try my best to answer questions. Go back to the objectives, make sure you can answer them. And if you have any questions, please email me.